What I do in the Vall d'Hebron Institute of Oncology, I am the director of the Translational Research Program, and I have a group that is devoted to study glioblastoma, which is the most common brain tumor. So what we do is we try to understand the biology of glioblastoma, trying to look for new therapeutic targets, biomarkers to stratify patients to be treated, mechanisms of resistance, but also we look for circulating biomarkers to try to characterize the tumor in a non-invasive manner. So we found that, for example, it's very important to detect circulating tumor DNA in the CSF of patients, because by sequencing the DNA that it is in the CSF, you can characterize, you can know what are the genomic alterations found in the brain tumor. And also we have, in a separate study, look at characteristics of the cancer-initiating cells that are present in glioblastoma and how these cells are regulated by a factor called LIF. This is free DNA. So what happens is the tumor, when it grows, some of the cells die, and then they release their DNA to the microenvironment, and then this DNA is washed out by the cerebral spinal fluid that is bathed in the whole parenchyma. And this CSF, cerebral spinal fluid, goes from the parenchyma down to the spinal cord. So if we have a lumbar puncture and we obtain a sample of this CSF, and then we capture the DNA that is present there, we can sequence it. And so we can see which are the genomic alterations of the tumor without having to do a biopsy, which, as you can imagine, is very invasive because you have to go through the skull and get into the brain. It's much more successful than assessing ctDNA, circulating tumor DNA, in the blood. So for some reason, brain tumors do not release so much ctDNA into the blood. They normally get it into the CSF. And then the concentration of ctDNA is higher because the amount of normal DNA is very low. So the ctDNA that you find is normally all coming from the brain tumor. And second, the volume is smaller than the blood. So that's why it's not so diluted. Yes, we are making a huge effort to try to translate these discoveries into the clinic, because we think that it's very important to really characterize the tumor in a non-invasive or relatively non-invasive manner, especially brain tumors, which are difficult to access, in order to improve the treatment, but also to try to know which is the prognosis of these tumors. We want to know whether the tumor is benign or aggressive, because this will dictate the type of treatments that we have to do. Of course, I mean, this is something that we think is very beneficial for diagnosis, so know exactly which is the tumor that you have in the brain. Second, prognosis, whether it is benign or aggressive. Third, monitor the tumor growth, whether it is responding or not. We know that the levels of ctDNA correlate with the burden, with the tumor burden. So if the tumor is reducing, you have less levels of ctDNA, and if the tumor is progressing, you have more levels. And then, obviously, if we are lucky and we see some of the genomic alterations that are druggable, that there is a drug that can be used, then we can also impact on the therapy. Our department is, as I said, a translational department. So what we want to do is translate as fast as possible the discoveries in basic research to the patient. We have a very nice multidisciplinary group that include many different disciplines: oncologists, pathologists, surgeons, and scientists, all together trying to help in understanding and trying to help in the brain in the treatment of patients. One of the models that we are using is what we call PDX models, patient-derived xenografts models. So basically, we obtain the sample of the tumors of the patients that are undergoing surgery in our hospital, and with this sample, we inoculate them into mice, so to recapitulate the tumor of a patient in the mouse, and then we can study the tumor in the mouse, and also we can use that model as a preclinical model to understand the mechanism of action of some of the compounds that are in development.